Number 7, use of embryology in phylogeny. First thing, we need to define what an embryo is. An embryo is an early stage of development of a multicellular, that means it has more than one cell, diploid, that means that chromosomes are in pairs, eukaryotic, it has a nucleus, and organism, you know the word. So it's how a organism will develop in the early stages. And what scientists have noticed is that by studying this, you can actually see that there are some differences, but there are also some similarities. And by comparing these similarities, you can then establish some more close or more distant relationships. 7.1, invertebrates larvae. First thing, I've put speech marks for invertebrates because, well, just like reptiles, it's not really a phylogenetic group. The reason is scientists nowadays tend to give names to groups based on what the organisms have rather than what they don't have. So invertebrates is very useful to describe a big group of organisms, but in terms of phylogenetics, it doesn't correspond to a proper group. So we're going to illustrate how embryology can be used to make some relationships, to establish relationships between species, and we're going to use the Bac exercise from 2018. So this is the Bac exercise, and the question is the following. Based on your observations of figure 1, suggest a classification in two groups of two species that are most closely related. Well, if we look at the adult forms, it's maybe not the easiest to establish which one would be closer, because for instance, Mytilus edulis, which is a mussel, well, has a shell just like Eulidia hyperborea, but you could argue that this one seems also to have some kind of shell. So maybe not obvious to know which one's which. Perhaps this part could remind you of this part. So when you look at the adult forms, it's not always obvious. We saw that, for example, skeletons, it works quite well, but in this case, looking at the adult forms is maybe not the easiest. So what we're going to do, we're going to look at the larva forms. Let's have a look at that. The larva forms, compared to the adult forms, have quite significant characteristics. If we compare Lepus ancirifera and Artemia salina, if we look at the global shape of the larva, well, there are two things which are striking, is that you've got like a circular center part, and you've got these kind of arms, legs, if we may say so, with these kind of perhaps fluffy parts at the end, or some kinds of solid hairs. I'm going to take the paintings off. So these two larvae are quite similar, and the same goes for the other two. We've got one circular shape, no, and well, what looks a bit like arms, none of that. Only one. So here we've only got one, and here we've got multiple, but there seems to be layers of very thin hairs. So we can say, by looking at the larva stages, we can make two groups. So B and C, looking at the larva stages, seem to be quite similar. So we could say that they're one group, and A and D, they're going to be another group. Very rapidly, question 2, briefly explain the use of DNA sequences and the expected results when comparing these four species. Well, based on our observations, it would seem logic that species C and B have closer DNA sequences. That is to say that they would have less differences, less mutations that are different, so very similar sequences. And the same would go for A and D, our hypothesis would be that the DNA sequences for A and D would also be quite similar. And now, if we compare the A and D compared to B or compared to C, we should be able to see much differences. So if our observations are correct, the DNA sequences should also give information that A and D are more closely related and that C and B are more closely related. 7.2, vertebrates, comparative embryology. So this time we're going to talk about vertebrates. Vertebrates is a proper phylogenetic term. It regroups a common ancestor and all the descendants. So this is a term that is properly used. So let's compare the embryology of these different species. So on purpose, I've shown you the more advanced embryos, and then we'll go back in time and to see the beginning of the embryonic development. So if you look at this third big part of the embryonic development of different species, you can start to identify specific characteristics, and you could almost guess which species it is. So number one, we have a fish. Number two, you might recognize something which looks a bit like a salamander. So we've got an amphibian. Number three, you can see that there is some kind of shell, a bit like a tortoise. Number four, you can see some kind of beak. It's a bird. Number five is a mammal, and it's a pig. And number six is a human. So at this stage of development, it's quite clear that there are some differences. However, if we go a little bit before, in this case, it's not as clear. You could imagine, you could perhaps say that, okay, this looks maybe a little bit like a fish or a salamander. Same thing for that. It looks a bit like a salamander. But for these four, you would really struggle to say which species it is. So that means that throughout embryonic development, different species go through similar stages. The way that these cells will organize will be very similar. And if we go even further back, it's even more striking. In this case, I would really dare you to be able to bet on what species is what. In this first step, they all look pretty much the same at the beginning. So that really means that from one cell to a few thousand or maybe a few million cells, the way the cells organize into tissues, into proto-organs, maybe not quite organs yet, it's very similar. So that shows that there is some similarities and that there is this idea that there is a common ancestor to all these species. Underneath your diagram, your Europaea document, you've got analyze the document. So you can write the following: the early stage of embryogenesis, which is another word for embryonic development, are similar for many species. This is evidence that they had a common ancestor. We saw that DNA, the genetic code, certain proteins, certain metabolic ways were common to all species. Well, embryogenesis is very similar for also quite a big part of species. So again, these are elements that show that many species have certain common ancestors. 7.3, atavism. What is atavism? You've got the definition in your booklet. So basically, it's when an ancestral trait or character reappears. So for many branches, many stages of different species, well, this character has been lost and then it reappears suddenly. We're going to see one example of this for you to understand. To illustrate atavism, we're going to look at the following example. First of all, I'm going to show you the classical development of a salamander. So we start off with an egg. It will develop into a larva. Then the larva will in turn have gills to breathe, legs, etc. Then we've got this fully developed form, and then this fully developed larva form will transform into an adult form. So this is the natural cycle for a salamander. I'm going to show you another species now, an axolotl. And you're probably thinking, but sir, why is this an axolotl? Could it not be simply a larva stage of a salamander like we've just here? Well, you're right, they are quite similar. But the difference is that this axolotl here can actually reproduce. So what's happened here? Well, this is a case of neoteny, which is one possible case of atavism. These larva stages, they are traits that most likely the common ancestor to salamanders, axolotls, etc. had. So what's happened is that in the development process of the axolotl, it's actually stopped in this larva stage. However, the different genes for reproduction were still activated. So you end up with a adult species which has ancient characters that the common ancestor had. Another example we can talk about is the fact that certain snakes have legs. This is a case of a reappearance of an old character. The common ancestor to snakes and lizards and other species had legs, and snakes throughout evolution lost their legs. And well, there are some cases where you can see that it's not a very long lizard. It's really a snake which has this reappearance of a nascent character, which is legs. So let's just have a quick look at the video. So that's another example of atavism. You might be wondering how such big changes can happen. The fact that the axolotl stayed in a larva stage, if I may say so, and that the snake had legs which appeared, if I may say so. Well, the answer is that it only takes a few genes for that to be possible. There are certain genes which will code for the organization of different structures. And if these genes are altered, or if genes which regulate these genes are altered, you know that in the beginning of the year when we talked about the lactose operon, for example, different genes can be regulated with repressors and activators. So if these genes which determine the organization plan are altered, if they have mutations, well, whole structures can disappear, or if there's a duplication, a whole new structure can appear. So I've got this document where you can see different and some of these genes which plan the organization, and what's interesting is that some of them have quite similar sequences in, well, you recognize the Drosophila fly we studied in the beginning of the year, and some of these genes are very similar to the ones we have. So again, this shows that there is a common ancestor to these species. And just to finish on this point is that these specific genes have a huge impact on how the embryo and the adult organism will be organized. To check if you've understood some of these concepts about embryology, atavism, there's this back exercise you can do, which is page 31 and 32 in your booklets, and it looks into this question: are cetaceans the ancestors of terrestrial mammals? Cetaceans being, well, dolphins, whales, etc. So I'll let you have a look at this to see if you can check if you've got these basic principles. Okay, well I'll let you have a look at this back exercise. To finish this video, I found a quite funny parody of Despacito, so a biology version. So I'll let you listen to that. Hope you enjoy it. That's all for this video. Goodbye.
See, one cell divide and decide on 1,000 fates. Did you ever figure how they know? We are built of modules combined in a planned out way. Each new piece must be told where to go. Evo, Devo, looking at the logic in the ways that we grow.